go. So the first 4 kingdoms, we're going to kind of jumble those in here in Module 3. And the reason is because we don't need to spend a lot of time on kingdoms, Monera, Fungi, Protista so much. Okay, we'll be spending most of our time on Protista today. Protista being the one exception to that. Okay, but the unicellular algae we'll look at today. Okay, just a reminder, on the 17th of May, we have a project due in this class, right? Y'all are going to be presenting a project. It is a PowerPoint presentation. You'll be standing up here and be doing this. So those of you in my history class, yeah, so history class is going to have 2 PowerPoints at the end of the year to deal with. One from marine biology and one from history. Okay, so we probably will have to, because of the size of the class, we might have to bump a few up to the previous week, and that'll work, especially if I have a student who, there's always a student who takes summer break early. Anybody know that if they're taking summer break early and they're not here the last week of class? So we'll bump you up to take, to present your project sooner than that. And so if, I don't know if y'all, some of y'all might be the type that say, let me present it and be done with it, kind of a thing. And the reason I'm bringing it up right now is because in your lab book, you're going to come across pages and it says project presentation or something. That's because apparently other teachers sprinkle them throughout the year. I don't know if anybody was ready to present a project at this point in time. Right, so we'll definitely do them in the second semester. If you want to do them sooner even than May, we can do that. Okay? All right, so let's look at Kingdom Monera. If y'all remember, I'm a microbiologist, so I like the bacteria. That's my thing, you know. So I like Kingdom Monera, but there's not much in the ocean to deal with, okay? These are prokaryotic, single-celled, microscopic organisms. Some people erroneously call them germs. They're not germs, all of them. Most of them are beneficial. Some of them do cause disease. Those are the bad guys, yeah. Give them a bad name. Marine bacteria are usually rod-shaped. They usually have cell walls. Keep in mind, they also have to deal with the salt. Just like we talked about last time, dealing with salt and how animals have to come up with a strategy to deal with the salt. So do the bacteria. All right, so their cell walls typically will do that for them. Most are heterotrophic decomposers. Okay, so heterotrophic decomposers. Who remembers what those were? Decomposer's easy, right? Breaks down that stuff. What's heterotrophic?
It eats other things.
Yeah, it's not making its own food, right? It's not making its own food. They're the best recyclers on the earth, so they recycle dead things. They mostly live in bottom sediments, kind of the stinky stuff. You ever been at the ocean? There's that kind of silty rather than sand, and it's sort of smelly, like in the bay area, for instance. That's the remnants of the bacteria in there. That's why it's smelly. All right, so let's see. There are 3 types of bacteria. This is one of your blanks, yes? Number 1, autotrophic bacteria are able to photosynthesize. Okay, so these are able to photosynthesize. All right, there are not a lot of them. There's one very important one that keeps being misnamed algae, but it's not algae, it's bacteria. We'll get to that one. The second group are the chemosynthetic bacteria. Now, chemosynthesis is similar to photosynthesis, except it's not using the sun for its energy, it's using chemicals. And those chemicals are coming from like cracks in the ocean floor. These are the deep ocean vents, we call them. And those chemicals are rising up through there, and bacteria are able to use those chemicals to make their own food. And number 3, the heterotrophic decomposers, like we said in the previous slide, they break down DOM. That's something we're going to come in contact with the rest there. DOM stands for dead organic matter, DOM. And it breaks that down into smaller molecules, making it, of course, getting it out of the way for one. Decomposers are important on our planet, in the ocean as well as the earth. All right, so 3 types, or 3 classes, I guess you could say, and we'll be looking, the vast majority of them are one shape, however, and that shape would be rod-shaped, or bacillus, the bacilli.
Are we supposed to be writing down
Apparently the things that are
parts?
underlined.
Oh.
But if you want to shorten it, shorten it. You don't have to write down everything I wrote. For heterotrophic decomposers, just say break down DOM, if you just remember what DOM is. And that chemosynthesis is like photosynthesis, but chemicals. Both chemicals, yeah. All right, so here's some rod-shaped bacteria right there. Okay, that's a very close-up view of them, probably a couple of thousand X, so we can see because they're very, very small. Okay. In fact, I don't even know if we're going to be looking at any bacteria today. But this one's the most important one, our autotrophic bacteria. Some of them perform photosynthesis. They have chlorophyll. So in other words, like a plant, they have chlorophyll, which is the green stuff which allows them to perform photosynthesis. They use light energy to make food, and the other ones are the ones that perform chemosynthesis. So that's what we're looking at right here in this picture is our chemosynthetic, because these right here are called tube worms. And tube worms are found at the bottom of these ocean vents. They're really crazy looking creatures. We didn't know about them for a long time because they're so deep in the ocean. And they live off the food that's created by those chemosynthetic bacteria that are down there. So pretty cool. But this is the most important one right here, the cyanobacteria. Okay, cyanobacteria, traditionally called blue-green algae. They are not algae because they're single-celled prokaryotic organisms. Okay, they're prokaryotic. Remember what prokaryotic is, right? We just did that, right? So our just has the DNA floating around, yeah? Right? There's no membrane-bound organelles inside, not the fancy, machinery inside the cells. So they're not algae, but they're called blue-green algae. It's tradition, so we tend to still call them that. Just keep in mind they are bacteria, cyanobacteria. They're super important. They contain green chlorophyll along with blue and red pigments. That's why they're kind of blue-green. The chlorophyll is contained in an unusual fold near the cell membrane called a thylakoid. Thylakoid, that's just some information for you. It's a little bit different than the way the plant holds it. Okay, the plant holds it a little differently in its little chloroplast, which is different for plants. That's another thing we'll talk about in this class. We'll talk about marine plants. Also, not that many marine plants. The vast majority of green stuff in the ocean is cyanobacteria, other algae, and like things like kelp. Kelp is not a plant. It's a algae. I think I have a picture of this thylakoid here coming up for you. Here we go. All right, so the importance of these, they are probably the biggest photosynthesizers on the planet. They also fix nitrogen. They make lots and lots of oxygen. People talk about, be careful cutting down forests. I'm not for cutting down forests at all. I'm for replanting forests. I love trees. But the vast majority of our oxygen is coming from our oceans. So really, we should be thinking about keeping our oceans healthy. All right, here's a picture of it. All right, so it looks like this. And does it show us those thylakoids are right? What's it pointing at there? Right, these things? Yeah, so these kind of sheets right here. And if you look inside the membrane, you can see they're kind of a fancy, different mechanism, but they are still going through the same process of photosynthesis, which we know that formula, yeah? Here's our nitrogen fixation. You don't need to know this. It's just that we're taking nitrogen and we're making it so that it's usable. In this condition right here, with our triple bond, it's not usable. It's all locked up, if you will. And over here, we'll get to NH3, and that's usable by other creatures in the ocean. All right. So this also shows the nitrogen cycle. You might remember this from Biology 1, and we talked about how the nitrogen's always being recycled. And keep in mind, when we breathe in the air, the 75% of the air we breathe in is nitrogen, right? And nitrogen is very important for the growth of green things. It's like a vitamin for green things. All right, so algae blooms. When floating in the warm water, we're talking about the blue-green algae now, the cyanobacteria. If they get too warm, they can multiply rapidly, producing dense concentrations of colonies. That's known as red tide. Has anybody ever experienced red tide? If you're going to the beach, or you're going to the coast, down at the coast of Texas, and you look up the reports and it tells you it's a red tide, don't go. It's not going to be any fun at all.
We accidentally went once.
Yeah.
We were there for like a whole weekend and we kept sneezing.
Oh, it's awful. It just, there's all these little spores that get in the air and they get in your eyes, they make you sick. People, start to feel, nauseated and stuff like this. So, but that's called red tide. They can't really predict it, but they can tell you when it's happening. So if you have a trip planned and they say, hey, it's red tide, I honestly would cancel my trip. It's that bad. You certainly can't get in the water, but even standing on the beach is not comfortable. So cyanobacteria colonies will release toxins, and that irritates humans, and it can actually kill fish. This is what they look like. Remember, they're bacteria. They're rod-shaped. They're green. Here are some poor dead fish from the bloom. You can see all that green. So it looks like pea soup there. Yeah, so notice it was called a red tide. The reason it's called a red tide is because a different type of algae, which we'll look at in a minute, its bloom is actually red. So, but it actually looks like a green tide right there, right? Okay. Oh, here it is from a satellite view. We're looking at a lake. This is called Lake. I don't know if anybody recognizes this lake. It's called Lake Atilan. Atilan? Anybody know that lake? Sounds
They're a
almost like,
volcano.
yeah, San Pedro volcano. I'm almost sounding like maybe Mexico. I don't know. Maybe España. I don't know. But you can see it all in there. That green in there. It's a bloom right there. And you know, typically we think about flowers blooming and it's a good thing, but when it comes to these guys, it's not good. And they'll actually end up taking over ponds. You may have seen that before, where a pond is just totally covered with algae on the top. It's not healthy for the pond to have the algae take over the whole pond. It's healthy for them to be in balance. Okay, so what you need for a pond like that is something that eats algae. Something that eats algae includes tilapia. They eat algae. They're vegetarians, in fact. Okay, this, I don't know if this is in your book. It's certainly not, you're not supposed to write this down, but apparently I found it interesting and I wanted to put it here. It's a type of cyanobacteria. They form these complex colonies. They're called mats of stromatolites. They can grow to be a meter high. What's that high? They're found off the coast of Australia and the Caribbean, and they look like this. So those are just giant mats of that cyanobacteria. Now, I'm not exactly sure if they don't look very green. So I don't know if it's just like the dead remains of them or if they're still performing photosynthesis. I know what'll happen is they'll actually float a little bit when the water comes up and the tide comes up. And so I don't know that they're attached to the ground at all. Yeah, but I'm thinking it kind of ruins the beach, if you ask me. Yeah, so. All right. So we're going to talk about Kingdom Pro. So that was Kingdom Monera, right? We only have really the one we are interested in, which is cyanobacteria, blue-green algae, okay? Now we're going to look at Kingdom Protista. And Protista is broken into two massive subkingdoms, Kingdom Protozoa and Kingdom Algae. All right, so the algae are the plant-like protozoans. They are not plants. And the protozoans are the plant-like Protista. The protozoans are the animal-like Protista. They are not animals. They're just animal-like. They tend to remind us of animals. Okay, so let's talk about unicellular algae. Kingdom Protista contains eukaryotic organisms, so that's what makes them different from the bacteria. Bacteria are prokaryotic. These are eukaryotic. They have a nucleus. They have all those membrane-bound organelles, all that. The organisms are divided into the plant-like, called algae, the animal-like, called protozoans, and phytoplankton are photosynthetic organisms that float in the water. One thing you'll need to remember for the rest of the year, when I say plankton, I'm referring to floating, okay? It floats. If an old stick is floating in the water, it's plankton. So anything, if you're floating in the water at that moment, you are plankton. So, but the ones, phytoplankton, are the photosynthetic organisms floating in the water. That would also include those blooms of cyanobacteria, okay? Those would be considered plankton.
Aren't jellyfish technically
Absolutely.
planktonic because
Yep.
they can't swim against the current?
They can swim, but they can't overcome the current. Yeah. If anything can swim but can't swim against current, they're plankton. All right, so some of them look like this. Very pretty. Yeah, green. Those are probably stained green right there, but they're just very pretty. Here's a close-up of one of them. We're going to look at this first group. So do you have you have a set now? We're talking the groups, different groups with different questions about each one? Yes? No? Doesn't say diatoms there?
It says diatoms, but it just says the phylum.
Okay, unicellular algae, diatoms, phylum. Okay, so then we're looking at a picture of a diatom. Diatoms are super cool, all right? And we're going to look at several of those in the microscope today. They are found in phylum Chrysophyta. Chrysophyta. That's Chrysophyta. Chrysophyta. The word crystal, you can hear the word crystal in there. That's because their cell walls are made of glass. It's so cool, all right? Their cell wall is made of something called silicon dioxide, SiO2, which is the same thing glass is made out of. Okay, so when I say they're glass, they're made of the same chemical that glass is made out of. So they have little glass walls. They're the greatest oxygen producers in the world. They come in many fascinating shapes, and they are fitted in parts called frustules. They have a top and a bottom. It's called a frustule. In fact, let me pull this out real quick. We're going to use this in a minute, but this is dish with the top and the bottom. So that's kind of like what a diatom is. A little glass dish with the top and the bottom called frustules. We'll use that in a minute. Okay, here's a picture of some of them. This is an electron scanning micrograph of them, so a very, very close-up picture of their little glass cell walls. All right. Oh, here are some more. Check them out. They're just beautiful. Here's another one.
It's like a kaleidoscope.
I know. I will tell you the colors are added by a computer, so they're not really that color. They're clear, like clear glass. Okay, so their locomotion. This is one of your blanks, yes?
Mm-hmm.
They are true plankton. So what's that locomotion mean? They are floaters. How do they move? By floating. True plankton. They stay near the surface. Why do they stay near the surface?
For photosynthesis. So,
What? Sun. Yeah, is that what you're saying? Is that what y'all were saying? Photosynthesis? Okay, I was looking for the word sun and I kept hearing. All right, photosynthesis. That's right. So they're staying near the top. There's another picture of a close-up one right there of its little frustule. Okay. They do eat by photosynthesis. How do they eat? Photosynthesis. They make their own food. They're autotrophs. And this is just showing you here their little chloroplast in there. Okay. With their mitochondria, their nucleus. Remember, they have membrane-bound organelles. So you've probably heard of diatoms, yes? Heard of them before? You ever heard of diatomaceous earth? DE? You haven't heard of diatomaceous earth, have you? Okay. We use it for an abrasive because you know what it's made of? Little bits of broken glass. Because it's made of the cell walls of these diatoms. So therefore it's glass. And we can use it also for filters in pools. I guess it works as a good filter. And it's good insect control because the insects crawl over it and they cut themselves up and they die of dehydration.
Wow.
It's also in rat poison. They feed it to rats, the rats eat it, it cuts their stomach, the other part of the poison has something that causes them to bleed and then they bleed to death from the inside. So yay for diatomaceous earth. It's all these little creatures. It's very dusty. It doesn't cut us. It doesn't hurt us. It doesn't hurt some like ants. It doesn't hurt because their exoskeleton is so strong that it doesn't really cut them. But worms and stuff, slugs, it works great on slugs. It totally just cuts them to shreds. Yeah, they just ooze all their insides out.
I've never felt so bad for slugs in my life.
I don't feel sorry for them. They ruin your
but I
butt.
don't wish that on them.
Okay, so we're going to watch about reproduction. Okay, so we're going to be looking at reproduction, and this is going to give you a lot of details of reproduction. We're looking at some asexual reproduction here. So watch this little fun video about pustules. Uh-oh, you know what? I forgot to turn the sound back on. Let me start over. Okay, hold on. I had muted it for my last class. And here we go.
In one mode of diatom asexual
Oh, yeah, he
reproduction,
calls them
a
diatoms.
single
They're
diatom
diatoms.
copies its genetic material and then splits in two. The top half of the frustule goes with one nucleus and the bottom half goes with the other. Using the genetic information stored in each nucleus, each half of the diatom then reconstitutes the part of the frustule it is missing, so that in the end there are two new diatoms present. Now let's look at these two diatoms. Remember, the original parent's frustule split in two. The top half went with the top diatom in this animation, the bottom half went with the bottom diatom. Each diatom then grew the part of the frustule it was missing. But look what happened. The new frustule came in underneath the original frustule. Now for the top diatom in the animation, this doesn't matter. The original parent gave it the larger portion of its frustule, so when the new frustule came in, the new diatom was the same size as the parent. Look at the bottom diatom, however. It got the smaller portion of its parent's original frustule, and the new frustule grew inside of that. As a result, this diatom is actually smaller than the parent. To see the practical effect of this, let's let these two diatoms reproduce in exactly the same way. When they're done, notice the variation in size. The top diatom is as large as the original parent because it got the largest frustule part. But look at the third diatom down. It got successively smaller frustule parts, so it is significantly smaller than the original parent. As several generations of diatoms continue to reproduce in this way, eventually some of the diatoms get so small they can no longer reproduce like this. However, there are other avenues of reproduction available to them, and they will be discussed in other animations.
Okay, which are coming right now. Okay, so.
As you learned previously, in one mode of diatom asexual reproduction, the successive diatoms produced can get smaller and smaller because of the way the parents split their frustules. One way a small diatom can fix this problem is to shed its old frustule, becoming a naked oxospore. The oxospore can then enlarge, and then it can form a new frustule. Now the diatom is pretty large again, and it can continue to asexually reproduce.
All right,
That's so
and,
cool.
yeah. And true sexual reproduction here.
Diatoms also have a mode of sexual reproduction available to them. One diatom can form a sperm, the other will form an egg, and when the two unite, they will fertilize, forming what is called an oxospore. The oxospore has all the genetic information of a diatom, but it doesn't have its frustule. To complete the process, then, it builds its frustule.
Yeah, and now the word frustule sounds like fun. Okay, there we go. All right. Is this our next group? And are we done with
What
diamonds?
did you want us to put down for reproduction?
Reproduction,
Frustule.
asexual and sexual, and frustule.
Oh.
Write down the word frustule.
I was gonna say, that seems like a really important word.
Yeah, so just, they have asexual and sexual mode. All right, so our next group, right, is our silicoflagellates and our coccolithophores, yes?
Yes.
You like those? Our silicoflagellates and our coccolithophores. All right, silicoflagellates have ornate silica cell walls, so kind of like a diatom, and it has a flagella and chloroplast. So if it has chloroplast, that's its mode of eating is what? Photosynthesis. And flagella is its mode of locomotion. Remember a flagellum? A flagella whips around like a tail and it swims, so it makes it motile. Coccolithophores have round chalky plates as their cell walls, and they make the water a really turquoise color, a beautiful turquoise color. Yeah, keep that in mind when you're swimming in those beautiful waters. There's creatures in there with you, and they're good.
So we guess we could assume that coccolithophores are in the Caribbean Sea, which is why they're that color.
Yeah, I would think so. Mm-hmm.
Pretty.
Yeah, and I'll show you a picture of one here as soon as y'all are done writing. Y'all having to write all that down? So I would just say they have, I would write down their cell wall. I would write they have silica cell walls, okay? And they move by flagella. They eat by photosynthesis. Here's a picture of our coccolithophore. Oh, it doesn't look like anything. It's got those little chalky plates.
Looks pretty.
Yeah,
Looks like a bunch of little shells.
right. And so keep in mind that's really small. I think there's flagella in there somewhere hiding. Maybe in those holes. Giving them that motility. All right, are we good? Someone's gonna have to give Ivy those notes. Dinoflagellates. Okay, it turns out I actually have some living dinoflagellates with me right here in this box. They're my pets. I brought them today. I don't know if they're gonna cooperate with me today, but we'll look at them under a scope for sure. All right, dinoflagellates are found in the phylum Pyrophyta. Do y'all see that P-Y-R-R-O, P-H-Y-T-A, Pyrophyta. Thank you very much. The old name Dinophyta is what they used to be called. Dinoflagellates, Dinophyta. They have unequal flagella for locomotion. They have a short flagellum and a long flagellum. The short one wraps around its middle. It's kind of cool. But the long one gives it motility. Their cell walls are made of cellulose. There's no blank for cell wall, but I'd go ahead and say put on their cell wall is cellulose. Cellulose is the same stuff that's in celery that makes it go crunch. Some of them have an eye spot, which is sensitive to light because how do they have to get their food? They are photosynthetic. They make it. They have very fast asexual reproduction, leading to an overpopulation. This is our red tide. This is our red bloom that's going to show up. All right, it's called red tide. And some of them perform bioluminescence. They're very, very cool. That's how mine might not cooperate with us today.
So would they be found in places like Australia, New Zealand, that have those really
I
pretty
think that's-
bioluminescent caves?
I think that's one of the chief places where they're found. Now, they're found everywhere. They're found in your pond water, dinoflagellates, but the ones that are bioluminescent aren't as common.
I always wanted to go to those beaches that have like bioluminescent water. Like,
It only happens at a certain time. It's not like all the
They
time.
have some in Korea. I could be completely getting.
Are we good?
Pastor, what do they eat? Was that the
Photosynthesis. Yeah, they make their own food. Okay, so this is what they look like. This is the flagellum that is for motility, and this is the one that wraps around them, like the middle. They have like a groove, and it wraps around the groove, and we think that has something to do with feeding. When I say feeding, I mean moving to get to the spot, like floating is I guess what I'm trying to say. To float, but these are giving it true motility. I don't know what that second one around the belly is for. You can see the flagellum here. See this one? You can see motility there, but the other one's just wrapped around. I don't know, maybe it unwraps? I don't know. Here's another picture of some of them. These are some that, I guess these are the flagellum here. These are the bioluminescent ones, by the way. We'll work with these later in the year, yes.
do you think that it could be like sort of like a life preserver?
Maybe to keep them up. That might be. If it moves, maybe it kind of keeps them moving up towards the sun. They need to be there. Oh, here's one of them bioluminescing. Okay, it's glowing right there. And here's a beach. We're gonna have a video here to watch. Okay. So I mean, I would love to see that too. That's like on my list. I just don't know how you time it. How do you know when it's gonna happen? Let's watch this.
May 2015. While the southern aurora illuminates the night sky above, the sea below produces a strange blue glow. Each wave causes a ripple of intense color. The animals in the bay notice it first. Wading birds are attracted to small crustaceans caught in the glow. Each movement alerts others to this rare spectacle. People gather to marvel at this once-in-a-lifetime event.
Look at that.
Oh, wow. Ah, that is amazing. I've never seen anything like this before in my life.
That's wicked.
It may look like something from Willy Wonka's chocolate factory, but the phenomenon is real. A mass bloom of microscopic organisms caused by a rare combination of climate and nutrients. Under this microscope, I've got a drop of ordinary seawater, and it's full of tiny organisms, invisible to the naked eye, called dinoflagellates. And if I disturb them in some way, they combine two chemicals in their body to produce a flash of light. Watch. Dinoflagellates are one of the biggest single-celled organisms known. They're 1,000 times bigger than bacteria. They're neither animal nor plant, but have characteristics of them both. And when conditions are right in the sea, as they were in Tasmania, they bloom in enormous numbers. Bioluminescent tides like this one are certainly rare. However, dinoflagellates are found in huge numbers all over the world. They're among the most widespread of all bioluminescent life. Wherever they exist, these single-celled creatures highlight anything that moves.
But why do dinoflagellates behave in this way? It's certainly not to entertain us, though it obviously does. Well, it could be that it's a kind of burglar alarm. That when a shrimp or some other animal that feeds on the dinoflagellates by filtering them out comes along and starts to feed, it is, in doing so, illuminating itself. So that attracts the attention of perhaps bigger fish that might feed on the shrimp.
Just as a flashing burglar alarm alerts the police to a thief, the dinoflagellates expose their attacker to its enemies. The shrimp is revealed to cuttlefish with fatal results. And so the cuttlefish can hunt in total darkness. But while the dinoflagellate's light can work in this way, it's still debated if that's why they do it. Whatever the reason, the magic created by their light can be one of nature's most magical spectacles.
That's closer, Mexico, right?
Bow-riding dolphins are revealed as dazzling outlines. Whenever these lights appear, the way life in the ocean hunts and hides is transformed. Perhaps dolphins are guided to their prey by the light of the dinoflagellates. Only now has it become possible to film these scenes with such clarity. But every night, spectacular light shows like this play out somewhere in the vastness of the oceans.
Okay, good. Stopped on a dark slide. All right, so these are my pet dinoflagellates in here. Okay, they're in this glass deal. All right, so I have them. So the question is, I've been feeding them, so they're in this glass deal here. So I'm gonna come over in the dark part of the room, just in case they do it. All right, come on, guys. Yeah, I'm not gonna do it. Okay, so the reason they're not glowing, I promise you they do. They do. Every night I swirl them, and I'm like, oh, it's because they're on a circadian rhythm.
Oh.
And so they're like asleep right now, or something. So they didn't do it, but they totally glow this whole orb up. And I have to feed them, I have to put them right through here. We are gonna though stick a pipette down in there and see if we can see them swimming around in our scope today. So I did bring, I realized that they probably wouldn't glow for us, but if we had this class in the evening.
Circadian rhythm is a real thing.
It's a real deal. Yeah, they know when it's day and they know when it's night. Yeah. So, all right, that's red tide, right? So you can see there that when the dinoflagellates overbloom, then they end up with actually a red-looking one. So that's why I got its name, red tide. Okay, I'm gonna turn the light back on. Okay, so just prepare your eyes because I did make it darker in here. So, okay, there we go. Our next group are our protozoans, our marine protozoans. Okay, and these are the ones, look at them over there, they kind of look a little bit like little shrimpies or something like little animals, right? Okay, zooplankton are tiny floating organisms that include protozoans and some small animals, okay? So just keep in mind that our protozoans are in this group, but so are some, larvae of different creatures and stuff like that. But for the protozoans, we're just considering them in our talk today. All protozoans have a mode of locomotion. That's how they are differentiated. Haloplankton zooplankton or holoplankton. If you say it holo like that, it means you can remember it means whole. And they live their whole lives as plankton. They're floating from birth to death. And then meroplankton live only in their larval stages as plankton. So those are typically some of the animal creatures, non-protozoans typically. Little crab larvae and even fish larvae to some extent. So mero means part, a part. Our foraminiferans, our next group, they are in phylum Sarcodina. There's the old name for them. Ready? Sarcomastigophora. Sarcomastigophora. Anyway, Sarcodina is better and easier. So go with Sarcodina. That just reminds me that typically, scientists will go along and change things from time to time. And so sadly, if you go take marine biology in college somewhere, these names might all be changed according to their classification system. Okay, if they use domains instead of kingdoms and stuff like that. They resemble amoeba, so that's how they move around. They move around with the use of pseudopods. If you remember, amoeba are the ones that kind of ooze their way around, and they stick some of their cytoplasm in one side of their cell, and it causes them to move, making a little, pseudopod means false foot, making a little false foot that's going to be crawling over one direction, if you will. They have a cell wall made of calcium carbonate. It's called a test. Calcium carbonate you could think of sort of like a shell. A lot of shells are made of calcium carbonate. And it's made of, that calcium carbonate is CaCO3. It's very chalky. Okay, it's very chalk-like. Like if you think about a shell that gets ground up or something. So we see some of the leftovers of these guys, some of their dead bodies from the tests. The tests of them will collect up over time as they die and fall to the ocean floor. And this right here is called the White Cliffs of Dover over in England. And those are really, really tall. You can't see here. I think that's like a lighthouse over there or something, or a castle. It could be Dover Castle over there. Anyway, this is one of the tricky things for people who believe in the creation model, because where did those guys come from? Why are they there? So there has to be lots and lots of these little guys who have died, right, with their little shells that have collected up. They're microscopic, so that takes billions and billions of them. And how they get so high up? So I would think some sort of mass earth movement, some sort of earthquake pushing up a plate or something like that. But this is one of those ones when you go, well, gee, I don't really have an explanation. It seems like it would have maybe taken millions of years for something like this to happen. I don't know. I'm just being honest with you that this is a sticky subject for creationists. Eating and reproduction. They trap small creatures in their pseudopods, okay? So they eat by, they eat diatoms and zooplankton. So how would you say? They trap them in their pseudopods. You can see right here, these little pseudopods, they're sticking out little sticky, strings and catching those little creatures. And they have sexual and asexual reproduction modes. So notice they are not photosynthetic, right? They have to eat. They're heterotrophs. All right. Our next group are the radiolarians. Yes, is that right on your book? Mm-hmm. Okay. Our radiolarians, that's a close-up picture of one here. They're in phylum Sarcodina. They are plankton with cell walls of silica, similar to our diatoms, right? Silica. They're usually spherical and have needle-like pseudopods. I'm gonna try to do a little demonstration for you today about how a pseudopod can help slow sinking. So their locomotion is going to be put on their pseudopods, yeah. But they're needle-like pseudopods. And it really is not, they're not moving so much as they're not falling too fast. And they're waiting for something to lift them up and uplift them, like an upwelling or some temperature change or something like that to kind of carry them back up to where the food is. Oops, sorry. So does everybody have everything they need from this slide? Pseudopods. It's just when I say pseudopods, it reminds me of a amoeba, which is like a fluid type of pseudo. These are spikies.
It's like little tiny swords.
Like swords. Like what?
Swords.
Yeah, like a sword. Eating and reproduction. Pseudopods help track food. So they use their pseudopods to track food, similar to the last guy we talked about. And they have asexual and sexual reproduction. All right, our next group are called the ciliates. Cilia is a little like finger projections like this, you know, like little hairs, if you will. That's why they're called that. They belong to phylum Ciliophora. They're covered with cilia for locomotion and feeding. So their mode of locomotion, cilia. How do they eat? Cilia will push stuff into their little mouth groove area. And they have asexual and sexual reproduction. All right, we're going to watch a little video on these guys.
This is microscopic video of ciliates that are very similar to marine ciliates. Like marine ciliates, they are trumpet-shaped, and their cilia are at the mouth of the trumpet. The cilia are small enough that you can't see them without the aid of a stain, which kills the microorganisms, which would make for pretty boring video. Nevertheless, you can see that these guys are moving around, and they do so by beating their cilia. They not only use their cilia for locomotion, they also use them for feeding, sweeping particles of food into their bodies for digestion.
All right, and I have this, I don't know if there's any sound on this one, but it shows them. Look at the little cilia up there. So here comes something, I think they're gonna try to catch it, and boom. Just ate whatever that was, and it's in their body now. Did you see it? So now these look like they're attached to something. So we'll find that in the marine environments especially, they'll attach them, because the marine environment is so brutal with waves and whatnot. So it's hard to get microscopic pictures like this because since they're moving, you know, they're going in and out of, you know, they're. Okay, there's whatever it's going to eat, I think. Okay, that's all I have for that. All right, so we're going to be doing some microscopy today. The problem is I have all my slides here, and I'm having trouble making sure that I have everything you're supposed to draw. So if we end up having to draw off of here, we may have to do that. So here's our foraminiferans. There's one that we saw previously, and we might draw that one. Here's a dinoflagellate, but I do have those living, so I'm going to put those. I'm going to have my dissection scope up here at this table because it needs to be plugged in. I'm going to bring a scope to all of you at the table that has battery operation. We'll be looking at some slides. This is a radiolarian. I hope I have that. It's very cool looking. Here's another radiolarian with its spikes gone. I think it's probably just the test of it. So it's just the, yeah. All right, so I'm going to bring around a scope.
Are you supposed to do anything here, or is it all?
It looks like it's all on the next page, yeah. I might have you on that blank spot on that first page. I'll show you the little demonstration I'm going to talk to you about, and you might want to draw that. All right, so I would like y'all to take these scopes out of their boxes and pay attention how they go back in because we're going to have to put them back up too, okay? And they each have their own special box, and they're labeled with letters on them, A, B, C, A1, A, blah, blah, blah. There's at least one of them that's a little persnickety. we'll do our microscope work, and then we'll do my little demonstration and try to find our living creature.
Casper?